Okay, tonight we're going to cover cell division, meiosis, and a little bit about DNA structure and DNA replication. So I'm going to start with cell division or mitosis. And just as we get started, I wanted to point out that this is a karyotype, which essentially means a cell has been taken and all of the chromosomes that are in that cell have been labeled with a kind of fluorescent tag so that we can visualize all the chromosomes. And then with a computer program, so this is sort of what the chromosomes look like, right, once they were stained under the microscope. And then a specially trained person called a cytotechnologist has identified which chromosome is which based on its length and the way that it's stained with the different fluorescent labels. And so they have been lined up in this box. So that's the chromosomes there. They've just been put in a particular order from chromosome 1 all the way to the sex chromosomes. And this is what's called a karyotype. So essentially it's the arrangement of the chromosomes that are found in a cell. Now there's a few things that I want us to notice about the karyotype. Okay, the first thing is that this is a human cell, so as humans we have 23 pair of chromosomes, and 22 of those pairs are homologous pairs, meaning that, for example, if we look at chromosome 1 here, we see that there are 2 chromosome 1s, so one was inherited from the mother, one was inherited from the father, so even though the DNA sequence that's found in those chromosomes is not identical, the same genes are found on that chromosome. So they may have inherited one form of a gene from mom and one form of a gene from dad, but it's the same genes that are found on that particular chromosome. So notice that from chromosome 1 down to 22, the chromosomes get much smaller, right, as we go up in number. And the last set of chromosomes, which for this particular karyotype, this is a female because it shows 2 X chromosomes. If it was an XY, there would be one much larger chromosome than the other because the Y chromosome is a fairly small chromosome, which makes sense if you think about it because you can survive without a Y chromosome, right? Us females are surviving and we don't have a Y chromosome. So the Y chromosome is essentially the additional male genes that are found in males. So that's a karyotype. And I just want to make sure we understand that there's 23 pair of chromosomes. And that means that a human cell is what we call diploid. Okay, that my device is not doing very well today, but it's diploid. Diploid just means two, they usually abbreviate it as 2N, which means 2 copies of each chromosome. Now, as we talk about meiosis a little bit later tonight, we'll mention haploid. Well, haploid means only 1N. So in other words, it would be half, only one of these for each chromosome would be represented. There's our karyotype. Now let's talk just a little bit about DNA, genetic material, chromosomes, chromatin, all those different terms and how they're related to one another. So DNA in its strictly just deoxyribonucleic acid form exists as a double helix. So a double helix means helix is like a spiral shape. Okay, so if you took a ladder, hold on a second here. Okay. If you took a ladder like this, okay, and that was the sides of the DNA, and as the rungs go across, that's what's holding the 2 strands together. And if you took that ladder and then you twisted it, what you would end up with would be, these would be like the sides, right, of the ladder, these that have been twisted. So this is the state of DNA, okay? When you take that DNA molecule in a cell, what happens, there's not enough room for that to just be unwound and loose in the cell. So it has to be packaged so that all of those base pairs of DNA can fit in the cell. And so they get wrapped around some proteins so that they can be sort of packaged up in a more compact form. So this shows you that that DNA molecule then is being wrapped around. What's inside of this is a protein, okay, called a histone. That's the protein, okay? And there are actually 8 histones that come together, and the DNA will wrap around that, and the 8 histones plus the DNA together is what forms what we call a nucleosome. So in other words, all the histones with the DNA, one of those would be a nucleosome. The next one would be a nucleosome. So all of the purpose of that is wrapping the DNA around the proteins so that it is more compact or condensed to fit inside the nucleus. Now, this is DNA, okay? When we get to this here, we don't just have DNA any longer. We have DNA plus protein, right? It's wrapped around those proteins. That is what we're going to refer to as chromatin. So chromatin is DNA in a particular state. It's in this state where it's loosely wrapped around these proteins. Now from there, it can get further condensed. So in other words, those nucleosomes are going to further wrap around additional proteins to even condense the DNA further. It's still called chromatin, okay, at that point. It's still DNA wrapped around protein. It's more condensed than the one before. But the most condensed that the DNA gets is in the state of a chromosome. So in other words, this whole piece here, which are the nucleosomes wrapped around additional proteins, would be found as inside a piece of one arm of this chromosome. So the chromosomes are the most condensed form that DNA exists in. So the way I want you to think about this is, in the cell, the DNA is the genetic material, right? The A's and the C's and the T's and the G's, the orders of those, that's important. But it's not just completely loose in the cell. It's either going to be wound and compacted in the form of chromatin, or it's going to be wound tighter in the form of chromosomes. So the least compact or the least condensed would be chromatin, and the most condensed or the most compact would be when it's in the form of chromosomes. Now, we're going to talk about the cell cycle in just a second, so I'll repeat, but just to make sure we're clear. In the cell, the time that you see DNA in the form of a chromosome is only during cell division. So normally in a cell, when it's just going about doing its cell duties, right, the DNA exists as chromatin. So it's not as condensed as it would be during cell division. Now, why does it need to be condensed for cell division? Well, because we'll see that these chromosomes need to get shuffled around, right, in the cell and taken to the appropriate location before cell division. And it's easier to do that when it's condensed into this chromosome type structure. All right, let's talk about the cell cycle now. So cell division is going to be our focus from this point forward, but cell division only makes up a small portion of what the cell does its lifetime. Okay, so this whole circle, okay, from beginning to end, all the way around, represents the cell cycle. I'm sorry, my computer must be, the internet must be slow. My writing is not working out too good tonight. All right, this is the cell cycle. Now, this small piece right here of the cell cycle is cell division or mitosis. All the rest of it, the cell is either just doing its job as whatever type of cell it is, or it's in preparation for cell division. All of the rest of the cell cycle besides mitosis, besides the cell division, is called interphase. So interphase includes G1, it includes S phase, and it also includes G2 phase. Now in interphase, the first portion, G1, is so G for growth or gap. What's going on? Okay, the cell is growing, it's performing its normal functions as a cell. Then the cell reaches S phase, okay, which means DNA synthesis. What does synthesis mean? This means replication. So this is when all of the DNA is going to be copied. Okay, so cell division simply means one cell has to copy everything and then divide and split that into two separate cells. So we have to be able to make a copy of every chromosome, right? We have to have duplication of all those. So we have two complete sets so that they can be divided up among the two daughter cells. So DNA synthesis, I want to make sure I point this out. DNA synthesis occurs well before mitosis begins. The DNA is not copied or replicated in mitosis. It's copied or replicated during interphase. All right, and the last part of interphase, G2, which stands for either growth or gap2, however you want to think about it. This is going to ensure that all of the proteins, all of the organelles, everything has been duplicated and is ready for the process of cell division. We'll talk in a minute, there are checkpoints that make sure the cell is ready to progress to the next stage before it does so. Then we have this small piece of the pie, which is cell division. This is when the cell actually gets split, right? All of the DNA, all of the organelles, all of the cytoplasm gets split into the two daughter cells. Now, I'll sometimes just say mitosis as the cell division component. Mitosis is really keeping track of the chromosomes or the DNA to make sure that both daughter cells get their full, complete set. Cytokinesis is essentially the division of the cytoplasm and everything like the organelles that are in the cytoplasm. So these work together, right? A cell doesn't just divide its DNA or just divide its cytoplasm. It all happens, right, in the process of cell division. Okay, what is the goal of mitosis? So let me and if you have questions, please make sure you let me know, put them in the chat box or something. Okay, so we're going to talk about two types of cell division tonight. Okay, I'm going to hope that this works a little bit better. Mitosis and meiosis. All right, what is the goal? Let's talk for just a second about what is the difference between these two cell divisions and what is the goal of each of them. So mitosis, okay, you get two daughter cells and they are identical to each other and they are identical to the parent cell. Okay, so what would be the purpose of this? Well, when there's repair that needs to be done, when there's growth that needs to occur. Cells get old and need to be replaced. So this is just a normal process that cells go through or tissue goes through to replace or to repair cell tissue. Okay, exact copy, you get two daughter cells. You're starting with diploid. Remember we talked about diploid means two in or two copies. I'm going to put two in here. Two copies of every chromosome. So for humans, we have 23 pair of chromosomes. We get one of chromosome 1 from our mom, one of chromosome 1 from our dad. So 23 pair means we have our diploid number, right, is 46 because 2 times 23 is 46. We're starting with a diploid cell and we end up with two diploid cells. So that's the overall process of meiosis. Now, we go through several phases of mitosis, but it's one division. Okay, one cell divides through one division into two. All right, now let's talk about meiosis. Meiosis has a different function or different reason that it occurs. Meiosis only occurs for the process of producing gametes. Okay. What is a gamete? An egg or a sperm. Okay, the only time that meiosis occurs is in the production of these cells. Now, recall that an egg and a sperm will come together, right, and will fuse, and the first new cell is going to be called a zygote. The egg and the sperm, if they were both diploid, then when they combined, now you wouldn't have a diploid cell, you'd have a tetraploid or a cell with 4 copies. Well, that's not good. We want the new organism to be diploid. So you have to have a haploid. I'm going to represent this with N. N means 1 copy or haploid. And you have a haploid sperm. Okay, so when the haploid sperm and the haploid egg fuse, then you restore the diploid order, right? So you have a 2N or a diploid zygote. So the whole purpose of meiosis is to produce haploid cells. Or you could say it a different way, the whole purpose is to start with a diploid cell and cut the DNA or the genetic material in half. In order to do that, what happens in this process is you actually get 4 daughter cells that are haploid, but they are not genetically the same, right? They're different. Obviously, they're going to be haploid. The starting cell was diploid. They're also not genetically identical to one another. So when we get it to meiosis in a minute, we'll talk in the details about how that works, but I want to make sure you understand the basic function of both of these cell divisions, so they're for very different purposes. All right, let's look at a chromosome close up for just a second and examine it. Now, a chromosome with one arm like the one I've just drawn here, this that I've drawn is a chromosome. Now, after a chromosome has been duplicated or replicated, in other words, everything's been copied, okay, it will look like the X that we recognize as a chromosome. So in other words, if you think about it like this, okay, if this arm was the original one that was over here, then this other arm is just an exact copy. Okay, so what we have here is a duplicated chromosome. So this chromosome, it has been through the S phase. One of those arms has been copied so that we have 2 identical arms. So if you look at it this way and you sort of split it down the middle, okay, those are identical copies. Therefore, this one and this one are called sister chromatids because they are in fact identical. Okay, this whole area is what we call the centromere, okay? And what we're going to see is when we talk about cell division, we talk about what's called the spindle in the cell, which is essentially made up of parts of the cytoskeleton called microtubules that attach to the centromere, and that's what's going to move the chromosomes around to the appropriate part of the cell during cell division. So notice it shows the centromere, it shows the microtubules that are part of the spindle, and then it shows these specialized protein structures called kinetochores that are part of the centromere, and that's where the spindle is actually attaching on the chromosomes. All right, let's look at the phases of mitosis. So keep in mind that this is a process, right? So the cell begins mitosis and it goes through the process until the end of cell division when it has become the 2 daughter cells. So it's not like the cell is saying, oop, I'm in prophase, I do this, 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 oop, now I'm in prometaphase. These are phases, right, that we use to describe the process, okay? So what is important to know about these phases? Well, prophase is the first phase. And there are a few things you need to know that occurs in prophase. So the first thing you're going to want to notice or be able to recognize is that the chromosomes condense and become visible. So remember, what's before mitosis? Well, right before mitosis begins, the cell has just been in interphase. Okay, remember what I told you about the state of DNA? It's chromatin, right? It's loosely wound as long as it's in interphase. The only time a cell has chromosomes is during cell division. So the DNA exists as chromatin, this sort of, if you look at it under the microscope, it just looks like a the nucleus just has lots of thread-like looking DNA in it. Okay, it's not as tightly wound as a chromosome. But when it enters cell division, the DNA begins to condense into these chromosome structures. So you can start to see the chromosomes in prophase. What else happens? You can begin to see the spindle fiber. So the spindle just means that's the cytoskeleton, part of the cytoskeleton, that's going to be used to manipulate these chromosomes during cell division. So you see the spindle forming. You also notice that that envelope around the nucleus begins to break down. Because remember, the DNA is in the nucleus, right? Enclosed by the nuclear membrane. You also see that the nucleolus, which is the darker structure inside the nucleus where the RNA is produced, it disappears. Now moving on, we get to prometaphase. So prometaphase is sort of like, if you think of mitosis as having four phases, prophase, metaphase, anaphase, and telophase, then prometaphase would be that I'm in between, right? I'm in between prophase and metaphase. So at this point, what do we notice about prometaphase that's moving forward from prophase? Well, we notice that the spindle is attaching, right, to each individual chromosome at this point. So you can see these blue lines that are radiating out. This is what we call the spindle or the spindle fibers, okay? And these are beginning to attach to the chromosomes, okay? Now you'll also see that what we call centrosome, so this is sort of the area where the spindle fibers, right, are radiating from. In animal cells, that organelle, the centriole, right, there's a pair of centrioles that form the centrosome. In plant cells, they don't have centrioles, but they still have spindle apparatus that moves the chromosomes. All right, metaphase. As we move forward, then what happens? Well, notice what you see is the chromosomes have been moved so that they're lined up single file in the middle or the equator of the cell. So the spindle is attached, right? That's what's manipulated the chromosomes to line up in the middle of the cell. All right, moving along to anaphase, this is when we see the first separation of the sister chromatids. So in metaphase, right, we have the chromosomes lined up in the middle. The sister chromatids are still connected, right, at the centromere. In anaphase, what happens is the spindle fibers shorten, right? And with that, they're pulling apart the sister chromatids. So here is one arm, right, of a chromosome, and here's the other arm. They've been separated. Once they are separated, they're no longer called sister chromatids. Now they're called sister chromosomes, okay, because they are a chromosome fully in their own right. If they are attached together, they're called chromatids. Once they're separated, they're called chromosomes. All right. Moving on, the last phase of mitosis is telophase or telophase. And I want to make sure you understand that telophase is happening in conjunction with this last portion we're going to talk about, which is cytokinesis. So we'll get there in a second. But what happens in telophase? Well, first of all, those chromosomes are going to basically arrive, right? Get to opposite ends of the cell. So it started in anaphase, they started separating. In telophase, they're going to arrive there. And then what the other things that happen are essentially the reverse of prophase. So the spindle goes away, breaks down, the nuclear envelope comes back, and the chromosomes then are going to begin to decondense. So in other words, go from chromosomes to chromatin. Now, all of that was dealing directly with the chromosomes, right? The DNA, the genetic material of the cell. Well, the cell has cytoplasm, it has organelles, it has other things that have to be taken care of. So cytokinesis is essentially that, the separation of the cytoplasm and everything else that's in it. And it happens differently whether we're talking about an animal cell or a plant cell. So an animal cell, it's called a cleavage furrow, and it looks like this picture right here. It just looks like it begins to, the cell membrane or the plasma membrane just begins to pinch in as if you tied a string around it and just pulled the string tighter and tighter until essentially you've created the two separate cells. Okay. In a plant cell, because of that rigid cell wall, it doesn't happen that way. Instead, there's something called a cell plate that vesicles begin to bring in components of a new growing cell wall in between that we call a cell plate, and eventually, as those vesicles all join, we have the formation of a new cell wall. So this is just a little bit better look at the two types of cytokinesis. So you see in animal cells, the cleavage furrow, it's like a string or a ring that you tie around the middle and you end up with your two cells, whereas in plant cells, we have these vesicles that bring in portions of the cell plate, right? And eventually we get our two individual plant cells. All right, so I did tell you that there are some checkpoints, right? As far as the cell cycle goes. And that's to make sure that a cell doesn't begin to enter into mitosis if it's not prepared to do so. So notice that we have a checkpoint at G1 to make sure that everything that should have happened there has happened. We also have a checkpoint at G2, okay, make sure that all the DNA has been replicated and all the proteins that we need have occurred. And then we have a checkpoint during mitosis to make sure that in fact the chromosomes are arriving right where they should be. Otherwise, there's going to be a repair or some work to be done in that cell before it continues on in the cell cycle. Okay, before we move on to meiosis, I want to talk to you a little bit about cancer. So I did record a video for you, so if you want some more information, if you don't get quite enough tonight, I definitely recommend watching that video. It's posted in the lesson folder. But what is cancer? Well, cancer really is just uncontrolled cell division or uncontrolled cell growth that's outside of the cell cycle. And so this causes lots of different issues. One way I want us to look at how cancer develops is I want us to look at cell cycle control. In other words, there are controls that help a cell either enter into cell division or prevent cell division, okay? And we have something like an on switch and something like an off switch. So let's look over here and let's say this is like the on switch and this is like the off switch. And in other words, they're sort of working antagonistically, but really as a team to say, if there's more on switch, then the cell will continue through cell division. If it's turned off, then the cell won't continue through cell division. So on the on switch side, we have genes called proto-oncogenes. Okay, and these are normal, healthy, right? These are there for a purpose so that when cell division is needed, these will promote the cell into cell division. On the opposite side, we have what are called tumor suppressor genes. And these tumor suppressors do the opposite. They say, wait a minute, we don't need any cells. We have a lot of cells in this area. It's not time for cell division, or this cell needs some repair, right? There's a damage to the DNA. We can't allow this cell to enter into cell division until this is repaired. So these are both the normal forms of cell cycle control. Well, cancer can happen a couple of different ways. So, for example, if a proto-oncogene undergoes a mutation, it can become what's called an oncogene. So normally there are going to be signals that have to happen in a cell to turn on these proto-oncogenes so that they cause, they're promoting cell division. An oncogene would be when it's turned on all the time, right? There is no off switch for this. Whether or not it's receiving the cellular signals that say we need cell division doesn't matter. It's promoting cell division even when it's not necessary. So this is a way that cells can accumulate mutations because it's not stopping and waiting for repair, right? There's uncontrolled growth happening. So that's one way. On the tumor suppressor side, we can have the opposite thing occur. So in other words, if a mutation occurs so that we no longer have an active tumor suppressor gene. So in other words, it's like we're missing the off switch. We're missing the brake pedal. Okay, so that the only thing that's occurring is we're getting our proto-oncogenes, but we're not getting the opposite tumor suppressor gene. So we're not getting a halt. Hey, we've got DNA damage, so we need to repair this before we enter. Now, one specific tumor suppressor gene that I want you to be aware of is P53. So in a great percentage of cancers, we find mutated P53 genes because these are tumor suppressor genes that are common, and if they're mutated, then they're not doing their job for checking for DNA repair or halting cell division. The other tumor suppressor genes that you may have heard of are the BRCA or BRCA1 and 2. These are the breast cancer, known as the breast cancer genes. So in other words, these are tumor suppressor genes that what are if you're wondering if you've inherited this gene or you're wondering what this gene means, if you have those particular forms of the BRCA, that means that essentially you're inheriting at least one mutation already. So it doesn't mean that you will develop cancer. It means that those particular tumor suppressor genes already have one mutation that has occurred. All right, now let's talk about, oh, that file did not come through well, so I'll get it in just a second. I'll redo it. So let's talk for just a second about some more about chromosomes for meiosis, because we're going to move into the meiosis portion of the lecture. So we talked about a duplicated chromosome. So what we see here is we have a red chromosome, right, that's duplicated. So we have a red sister chromosome, two red sister chromatids, right? And we also have a blue chromosome. So the blue has two blue sister chromatids. So this would be like, let's say the red chromosome represents the maternal copy that was inherited, and the blue one represents the paternal or the father's copy. These two together, let's say they're chromosome 1, okay, or chromosome 2. They're the same chromosome. It's just mom, the one you got from mom and the one you got from dad. Together, these are called a homologous pair or homologous chromosomes. Now, in meiosis, we're going to see that these form what you see here, which is called a synapsis. In other words, they come together, all four sister chromatids, which we sometimes call a tetrad, in the very first phase of meiosis. And that's important because this is where we're going to see a lot of genetic recombination occur, because the arms or the arms of these chromosomes are going to exchange just a little bit of genetic material with one another. All right, so this is what I was talking about. So let's make sure that we understand that what we're looking at here is something that we call recombination. We also call it crossing over, because that's essentially what's happening. And this does not occur in mitosis. It occurs in meiosis, and it occurs in the very first phase. It occurs in prophase 1 of meiosis. So here are our homologous chromosomes, right? This is the same chromosome. So whether it's 1 or 2 or 3, it's the paternal and the maternal copy of the same chromosome. They come together, all 4 chromatids, and there is a crossing over. In other words, one arm of one and one arm of the other exchange a little piece of information. So now at the end, you see that a little piece of this chromosome has been exchanged for this one. And what that means is the sister chromatids are no longer identical, right? If you divide this one in half, they were exact copies, but now they're not because you have a portion that no longer is identical. And so we'll see in a minute why this is really important for genetic diversity. In other words, every time this happens, you might get a little bit different exchange on the arm of the chromosome. And so we're never likely to have two eggs exactly alike or two sperm exactly alike partly because of this crossing over event. Oh, sorry. I need to just try to grab that other file really quickly. I'm not sure why it didn't come through, but hold on just a second. In the meantime, while I'm grabbing the file, if you have any questions, uh, just type them in for me. Okay, here we go. This is going to be kind of small for you to see tonight, but you can also refer to this figure is right out of your OpenStax chapter 11 textbook. So we went through each phase of mitosis, and I want us to go through each phase of meiosis also, and then we'll just generally talk about the overview of what does this mean. So, what I want you to see is meiosis actually has 2 divisions, right? We have a meiosis 1 phase where we go through prophase, metaphase, anaphase, telophase, and then we have a meiosis 2 division where we go through those same exact phases. Now, which means we have to identify, are we talking about prophase 1 or prophase 2, prometaphase 1, prometaphase 2? So let's go through and talk about what happens, what's important for us to know in general about these phases. Well, in prophase 1, as I mentioned to you, one of the most important things that's happening, and here they have it labeled as chiasmata, is the crossing over, where there's a little exchange right between the homologous chromosomes or a little bit of genetic recombination that happens. So the other things are similar to mitosis in that we're seeing that the chromosomes condense, right? Because remember, before a cell enters cell division, it exists as chromatin. We see the nuclear envelope begins to go away, okay? And we begin to see the spindle forming. But the big thing that occurs in meiosis here is the crossing over event where the tetrads, right, the homologous pair are going to come together and you get a little bit of crossing over. Now, prometaphase 1, as we move on, there's some similarities to mitosis in that essentially the spindle is attaching to the chromosomes, okay? When we move to metaphase, we have the lining up along the equator in the middle, like mitosis, except there's one very big difference. And the difference is in mitosis, the chromosomes lined up single file along the equator. In meiosis, the chromosomes line up in their pairs. So in other words, the pair of chromosome 1s you can see are there together, right? The blue copy and the red copy are still together. They're lining up in their pairs. Now in anaphase 1, the spindle is again going to begin separating, but in mitosis what we saw was the spindle separated the sister chromatids, right? It's essentially one arm of the chromosome went one direction, one arm went the other. In meiosis, in this first division, all we're separating are the pairs. So the duplicated chromosomes stay intact. We're just pulling the pairs to opposite ends of the cell. And then in telophase and cytokinesis, essentially we have the formation of, so the chromosomes arrive at the opposite poles and we have the formation of two, everything's divided into two daughter cells. Immediately, we begin the second meiotic division, okay? Now here we do not have another crossing over event. It only occurs in the first prophase. All right, moving on to prometaphase, we see that the spindle is attached to these chromosomes, and notice it's kind of hard to tell the way they've drawn the picture in telophase 1 and prophase 2, but you can see that it is still, in fact, a duplicated chromosome, right? They just sort of drew them flat, laying down on those phases before. So now the rest of this looks similar to mitosis in that the chromosomes in metaphase line up in the middle or in the equator. And in anaphase, we see that the sister chromatids are actually being separated, okay? And in telophase and cytokinesis, we have the completion of the migration of those chromosomes to opposite ends of the cell. And you're going to end up with 4 different haploid daughter cells, and none of them are going to be identical because of the crossing over event that occurred. Okay, so remember, the whole point of this, we started with 46 chromosomes if it's a human cell, and we ended up with 23 chromosomes in each of these 4 haploid daughter cells. Now, if we look at what's going on here, meiosis 1 through meiosis 2, I do want you to see, so in prometaphase 1, right, we have the homologous pairs, okay? Then in metaphase, they line up in the middle, and in anaphase they begin to separate. But notice it's the pairs that are being separated, right? The sisters are still remaining intact. So this member of the pair will go one direction, and this member of the pair goes the other direction, right? And those will ultimately end up in the 2 daughter cells. Now, in prometaphase 2, each cell now only has one of the chromosomes, right? The 2 daughter cells each have one. So in this case, with this one, as it goes through metaphase, now you see that these are eventually going to get separated, right? The sister chromatids like you see here in anaphase 2, and these are going to get pulled to opposite poles of the cell. So in the first division, it's the pairs that get separated. In the second division, it's the individual sister chromatids that get separated. Oops, that's not what I wanted to show you yet. Let me just get it really quick like. Okay. Now here's what I would like you to see. That, remember I mentioned to you that we're very unlikely to ever see 2 of the same two eggs that have the same exact chromosomal arrangement because of crossing over and other things. So right now we're just looking at 2 pair of chromosomes is what we're going to look at. So remember, humans are going to have 23 pair, right, that can get shuffled up. So let's say that when this goes through meiosis, that they line up in this way, okay? The blue or the paternal on one side and the red on the other for chromosome 1, and same thing, blue-red for chromosome 2. Okay, after the first meiotic division, then those pairs are going to be separated, and one daughter cell is basically going to have the maternal copy of both, right? And one daughter cell is going to have the paternal copy of both. And then when you go through, you end up with the 4 daughter cells. So according to this, right, you'd have these 2 would be identical and these 2 would be identical. However, what really happens with crossing over is this one would have a little piece of the red chromosome. This one might have a little piece of the red chromosome here. This one would have a little piece of the blue, right? Because we know they're going to exchange genetic information before those pairs are separated. So in reality, with crossing over, none of these are the same. But if we just take crossing over out of the equation and look and see, okay, we can actually have different arrangements just because of the chromosomes. Okay, so let's say the next time, okay, so look down here at this one, next time what might happen is they line up blue-red and red-blue. So if you go through that, then the egg or sperm, whatever being formed, right, look different than what they did up here. Up here you had both paternal copies, both maternal copies. Here you have a mix. So if you keep in mind that there are 23 different chromosomes that can line up either the paternal on one side or the maternal on the other, okay, that in itself is 2 to the 23rd power. That's how many different combinations you can get every time it goes through meiosis. And then if you add in the fact that every chromosome can have a crossing over event and exchange a little bit of genetic information, right? You're talking about billions and billions of different possible arrangements that can occur for the development of egg and sperm. Now, this process that you're looking at here, we'll talk about later with Mendel a little more, but this is called the law of independent, let me see if I can write this that's legible, assortment. Okay, all that means is however chromosome 1 pairs line up, maternal on one side, paternal on the other, does not in any way influence how chromosome 2, 3, 4, 5, 6 line up. So every time it goes through this, randomly they can be in a different order, which makes for lots of genetic diversity. Okay, now let's talk about DNA a little bit. I want to first mention, and I did make a video about this, so hopefully you're not bored if you've already watched the video, but, whoops, that doesn't look like a G. Okay, so Frederick Griffith did an experiment with mice with 2 different strains of bacteria, okay? And what he did was, he said he was trying to understand what is the key thing. At the time, they didn't know what is actually being inherited from one generation to the next. What is the key piece of genetic information that gets inherited? So that's what he was out to prove. Is it DNA? What is it that's got some capability to be passed from one generation to the next? So he took a bacteria that he knew something about, and he knew that there were a couple of different strains of bacteria. So we're just going to call them the R and the S strain for rough and smooth because of the way that they appeared. Okay, the R strain was not pathogenic. So I'm going to put over here R. Okay. Non-pathogenic. Okay, so that means the S strain is deadly. Okay, so in other words, if he injected the S strain into mice, they died. But if he injected the R strain into mice, they did not die. So what he did was he took the S strain, which he knew was deadly, and he heat killed. In other words, he heated it up to the point that those bacterial cells were no longer living. He injected that into the mouse, and he said, what happens? Well, it was no longer able to cause the mouse to die. So he knew he had killed those particular cells. Well, then he took the heat killed dead S cells, okay? So he heat killed them. He knew that they could no longer cause the mouse to die, and he incubated them with some living non-pathogenic cells. So these didn't cause death, right? So he took living versions of those with dead or heat-killed versions of these, and then he let them sort of incubate together for a while. Then he injected that into the mouse. And you know what he found out? The mouse died. So he realized there's something here in these cells that were heat killed that was able to be transferred to the living non-pathogenic cells that gave them the ability to now become pathogenic. And he called that a transforming factor because he said it transformed, right, one strain of bacteria to be something like the other. So he knew there was something there that was passed or transferred from one to the next. So that was important historical discovery about DNA, sort of set some groundwork. Now, another one that I want you to be familiar with is the Hershey-Chase experiment. Now, when this experiment came along, there were many researchers who thought that protein was actually what was inherited from generation to generation, and that's what carried our traits. But there were others who thought, no, we think it's DNA is the genetic material that's inherited. So this experiment set out to prove whether or not it was DNA or protein that was inherited. So here's what they did. They took bacteriophages. Remember, these are viruses that infect bacterial cells. And the way that bacteriophages work, they must inject their genetic material into the host cell. That takes over the host machinery and produces more virus particles. So they knew leading into this experiment, whatever is the genetic material is what's getting into the cell. So they wanted to find out what is actually entering into the host cell. So they took the DNA and they labeled it with a radioactive chemical that allowed them to visualize where it ended up, and they also did the same thing with protein, and they labeled the protein with a radioactive label. So they allowed the bacteria to incubate with these viruses, okay, grew it up, grew it in culture. So what this is, okay, is a test tube with some broth with bacteria growing, allowing the viruses to do their thing. Same thing here. And the reason they've drawn the picture this way, where they show you the bacterial cell at the bottom with whatever got inside of it in there, and the leftover virus at the top, is because they took these test tubes that had the cells growing in them and they spun them in a centrifuge at a high speed. What that does is the heavier the particle, they go down to the bottom of the tube and they form what's called a pellet. It's a little portion of the bottom of the tube that kind of, you get a collection of all these cells. And then the leftover liquid part is just called the supernatant. Well, they knew that the cell and whatever was inside the cell was going to end up in the pellet, okay? And the virus, the leftover virus particle, and all the liquid would just be left in the supernatant. So what they were doing is they said, we want to test the pellet. We want to see what's in the pellet. Is it DNA or is it protein? And they could test because they had radioactively labeled the DNA and the protein. So when they radioactively labeled the DNA, they saw that, yes, in fact, they did get that radioactive label inside the host cell. So the DNA made it in, which means it's the genetic material. Just to double check, they did the same thing with the protein. They radioactively labeled the protein, but the protein did not get into the cell. In fact, it was found in the liquid supernatant. So this proved that DNA was actually the genetic material of the cell and not protein. So in general, let's just review the structure of DNA together. DNA is essentially a macromolecule or a polymer that's made up of individual monomers. The monomer of DNA is a nucleotide, and this is one nucleotide here. Okay, a nucleotide is a sugar, okay, which is shown in yellow. Sometimes it's called a deoxyribose, okay, because that's the type of sugar it is. A nucleotide also has a base, okay, or a nitrogenous base. The base is one of these that you see here, C, T, A, or G. So there's only 4 types of nucleotides. There's a cytosine nucleotide, thymine, adenine, and guanine. And then a nucleotide has a phosphate group. So that's essentially what a nucleotide is. The reason that I haven't said uracil or U is because you don't find that in DNA, you find it actually in RNA. All right, the other thing I want to point out is there are different types of bases, pyrimidines and purines. The purines, you'll notice, have this double ring structure. Okay, adenine and guanine are those. The pyrimidines just have a single ring. And the way that DNA comes together, DNA is double-stranded, and the strands are held together by hydrogen bonding between these bases. And the bases always pair in one way. Adenine always pairs with thymine, and cytosine always pairs with guanine. So you always have one purine paired with one pyrimidine. In RNA, uracil replaces thymine. Okay, now let's look at how this works. So we just looked at one nucleotide, right? And one nucleotide we saw had a phosphate, which is here, a sugar, which is here, and a base, which is here. So that's one nucleotide, okay? Here's a second nucleotide. These two nucleotides, notice, let me erase so you can see better. Okay, these two nucleotides are bonded together. Notice that the phosphate group of this nucleotide is bonded to the sugar of the next nucleotide. So if we draw a line right here, this would be like one strand of DNA. So this is nucleotide 1, right? Bonded to nucleotide 2. It could keep going, right? There could be third, fourth, fifth in a long strand. That would make up one strand of DNA. Now on the other side over here, we have nucleotides. So notice here's a nucleotide, whoops. Here's a nucleotide bonded to the next nucleotide, right? That's its own strand growing, okay? So we have two separate strands. Now, in double-stranded DNA, those two strands are held together by what's called hydrogen bonding between the bases. So these dashed lines that you see here, that's hydrogen bonding. Remember I told you that adenine always pairs with thymine and cytosine always pairs with guanine. So you can see that this is a adenine-thymine pair, which makes, there's 2 hydrogen bonds between that pair. This is a guanine-cytosine pair, and we have 3 hydrogen bonds there. So that shows us that a GC pair is actually, the bond is a little bit stronger than an AT pair because we have 3 hydrogen bonds versus 2. So that's a close-up look. Now, what I want you to see is, if you imagine, right, that this would be, let's say this is the side of the ladder, this is the side of the, oops, I don't know what happened there. Okay, there's the side of the ladder. Here's the other side of the ladder, and this would be like the one rung, this would be like the other rung, right, that are holding the two sides together. And then if you took that ladder and you twisted it into a spiral, this is what you would have. Okay, so this is like a cartoon representation, right? So that the blue ribbon that goes this way, this would be like the side of one ladder, right, that's become twisted. And this blue ribbon is the other side of the ladder. Now the colored rings that go across, the red with the yellow, the blue with the green, that represents the hydrogen bonding between the bases. So that's this right here, right? That's holding those two sides together. So when we say double-stranded DNA, that's what we mean, two strands that are being held together by the hydrogen bonds. Now the reason that we say with DNA that it has a sugar phosphate backbone is essentially that's what makes up the sides of the ladder. So notice that it alternates, okay? Phosphate, sugar, which would be the phosphate, the next one sugar, and it would just continue on. So that's the backbone, meaning like the side of the ladder, okay? Now, we're going to talk just a second about DNA replication. Maybe I should do that. We're almost finished, promise, but I do want to talk to you just for a second about DNA replication, and then I have some biotechnology that I want to mention to you, and then we'll move on. But I do want to show you a little bit about DNA replication. Okay. All right. DNA replication, so when we talked about cell division, we said all the DNA has to be copied or replicated during S phase, right? If you think about the cell cycle, there's G1, which is a growth or gap phase, and then we have the S phase. Well, the whole goal of S phase is to make a complete copy of all the chromosomes or of all the DNA. So what we're going to talk about now is how does that physically happen? Okay, it's called DNA replication, and DNA is double-stranded, and both strands have to get copied. Now there's a lot going on in this figure, and so in a minute I'm going to bring up the whiteboard and we'll look at it in a little less detail. But essentially, if you look back here, this is the double-stranded DNA. So this is like the starting material, right? One cell, this is a piece of DNA in one cell or a part of a chromosome. So the goal of replication is to open up that double-stranded DNA and make a complete copy of both sides of that. And so you see several important enzymes that are needed to do that in the cell, and the first of those is the enzyme helicase. So helicase's job, you can see it's doing it well there, is to essentially unzip the double-stranded DNA so that copies can be made of both sides of that. Okay, we also have enzymes called DNA polymerase, and notice that they have a couple of different polymerases listed here. I don't care that you know exactly what DNA polymerase 1, 2, and 3 does, but what I want you to know is the job of DNA polymerase is to produce the new DNA strand. Okay, it's also to prevent errors and repair errors in that new strand, but it's producing the new copy of the DNA. Okay, a couple other enzymes we need to know about: DNA ligase and primase and something called topoisomerase. I'll explain to you ligase and primase in just a second, but topoisomerase is there to make sure we don't have what's called supercoiling of DNA. So you know that if you have something twisted and you pull, let's say you're pulling at this one end, right, to untwist it, you're likely to cause it to kink up at the other end. So topoisomerase makes sure that the super kinking doesn't happen by creating little cuts or nicks in the DNA so that it relieves some of that tension that helicase is putting on it to unwind the DNA. Okay, now I'm going to bring up the whiteboard so we can talk about this a little better. And I'm going to draw here what we call a replication fork. So in other words, this is where the replication is happening in the DNA. Okay, so I'm going to draw in black the original DNA, in other words, what we're starting with. Okay, so notice that it's opened up because the helicase enzyme has opened it up. So I'm gonna put here some interesting things we need to know about, which is the ends of DNA. So this is the 5' end, and this is the 3' end. Oops, that's a 3. That's how we designate DNA. This is going to be the 3' end, and this is going to be the 5' end. Okay, that's important because that's how we know what side or end of the DNA we're talking about. Now um, I'm going to show you in just a second how you know what end of the DNA it is, but I don't want to get off of this figure until we finish it, and then I'll go back. I promise I'll show you that. So let's just put in some nucleotides so we know what's going on. Let's say there's a C here, and an A here, and a T here, and a C, and an A. So we know that these strands, they have to pair in a certain way. So if there's a C here, there must be a G here. If there's an A here, there must be a T. Okay, this is an A, this is a G, right? Because we know they only base pair in a certain way. So these guys, I'll draw 3 in, are base paired together. These are paired together. These are paired together. Okay, that's what's holding the double-stranded DNA together. Now, as it's unzipped, let's put in some sequence here. CA, C, A, G, let's say, C, T. Okay. Well, we got to go on, I guess, to the end. Okay. So here, this would be complementary. This would be a G. This would be a T, right? We have to, because these used to be together, right? They're just unzipped because helicase has come along and done that. That's an A, by the way. T. Okay. Now, the DNA polymerase enzyme that makes the new copy is only able to read DNA in one direction and make the new strand in one direction. So I'm going to put down here DNA polymerase. That's the enzyme making the new copy. Okay. It reads the template. So in other words, it reads what it's supposed to copy in the 3' to 5' direction. So in other words, the new copy that it's making is made in the 5' to 3' direction. So what that means is we're going to have one strand that's called the leading strand, meaning it can be done continuously. And one strand's going to be called the lagging strand because as it opens up, it's sort of going the opposite direction of the way the DNA polymerase can read it. So it makes a little piece, it has to jump off. As it opens up, it makes another piece. So let me show you what I mean. So the DNA polymerase is going to be up this strand up here, we're going to call the leading strand because essentially it's going in the same direction that the polymerase enzyme will work. Okay, so the polymerase will be able to begin reading from this direction and it will be able to make a copy continuously. Now, before it can do that though, there has to be something that comes in, the enzyme called primase, has to make a tiny little primer of RNA. The polymerase enzyme cannot start from nothing. It has to have a primer to begin from. So I'm going to make the new copy, I'm going to draw in blue. Okay, so the polymerase enzyme comes in and says, oh, I can read this from 3' to 5', and I can make my new copy 5 to 3. So where primase has left that little RNA primer, it begins and it makes the complement, right? So where it reads a T, it puts an A. Where it reads a C, it puts a G, and it just continues to do that. And as helicase opens up the strand, so if helicase were to come along and open up more of this, it could just continue on, right, the entire length until it was completed. And that's called the leading strand because it goes continuously. Now, this other strand down here is called the lagging strand because essentially it's opened up in the wrong direction. Okay, so it still needs a primer. So as it's opened up, let's say the primase comes in and it makes a primer right here, and then DNA polymerase can begin to make its copy right off of that primer. But it's going in this direction, right? It can only read from the 3' to 5'. So it will add the nucleotides, right? The complementary nucleotides. but then it's done, right? It stops. So then it has to hop off, and as this is opened up further, then it can come over here, right, and jump on here and make a copy to there and jump off, and it has to continue hopping as it goes down um, that DNA strand. Because it's not continuous, these little fragment pieces are called Okazaki fragments. And at the end, that enzyme I mentioned to you called ligase comes along and completes that and ligates those little fragments together so that we have one continuous strand. Now at the end of replication, what you'll see is we will have two complete, right? The leading strand will be complete, and after all of the fragments are put together, we'll have a complete lagging strand. And these will separate, and what we will end up with is something that's called semi-conservative replication, meaning that we will have, of the two copies, each of the copies, oh, let me just erase this here. So the parent strand, right, had, let's say it had 2 to start with, okay? After it goes through replication, the new copy is blue, okay? What we end up with is one of the new copies with one of each of the parent copies, okay? Because one strand is copied. So this is called semi-conservative because we've conserved one of the original copies with one of the new copies, right? And this is what gets passed to each of the daughter cells, one new and one old. Okay. We're almost finished, I promise. We're going to revisit this when we get to the biotechnology chapter. I just want you to see that. When we talk about DNA sequencing, that comes up a lot today, right? That the sequence of this gene or sequencing the human genome, the way that that is done is it mimics DNA replication that occurs in the cell, but we use a special, what we call a chain terminator, okay, that's radioactively labeled so that we can see the length that that chain terminated. So if this is the sequence down at the bottom, if this is the sequence of DNA right here, GAATTC, if that's the sequence that of the DNA, of course we wouldn't know it if we're sequencing it, right? But that's what exists there. Then what that means is that that in this particular terminator, okay, it's going to terminate for T, but it doesn't terminate every T. We have some regular T's and we have some terminator T's. So as it goes on, there's going to be, every time that there's a T, sometimes it's going to get the regular T that continues on, and sometimes it's going to get the terminator T. And by the end of this process, that means we're going to have a strand this long, right, for some of them because it's going to stop at that T. And then we're going to have a strand that's this long because sometimes it's going to stop where that T is in this particular tube where the T terminates. In the tube where we have the G that terminates, sometimes we're only going to have a strand that's as long as this first G, right? It'll stop there and we just have that tiny one. But sometimes it goes all the way to the next G and stops. And so we have a tube for the T's, a tube for the G's, a tube for the A's, and a tube for the C's. And at the end of the day, every single C or G or A or T, there has been a termination at that point. And so it gets read by a computer, and the computer reads it based on the length of the strand. So in other words, the shortest length of the strand has a G for the termination, so it knows that that's a G. The next shortest piece has an A at the end, so we know that that's an A. And that's how sequencing is done. Now we're going to go over this in more detail when we get to the biotechnology chapter. I just wanted to make sure that you know that it's using the process of replication. We're just adding this terminator in so that we can see where it ends. The last thing that I want to talk to you about tonight is something called gel electrophoresis. And this is a gel, and those little white bands that you see on this gel are actually fragments or pieces of DNA. And the way that a gel works, okay, let me get a white. Actually, I'm going to get yellow. Okay, a gel is, this particular gel looks like an agarose gel, and agarose is a powder or a substance that's made from seaweed that's very porous, similar to the agar in your petri dishes. If you poured agar in your petri dishes, it's kind of a porous substance. These here that you can kind of see are what we call wells. And once the gel is made, okay, we have these wells that are made with little combs so that you have sort of an empty spot in the gel for every time there's a well. Essentially, that's where each sample is loaded. So the sample is loaded into the well, right? Each of these samples is loaded into the well. So this is your DNA sample that you want to know the size of, or you want to separate in some way. And then an electric current is applied through the gel, and we have the negative electrode up here, okay? And we have the positive electrode down here, and a current is applied through there. Well, it just so happens that DNA, because of that sugar phosphate backbone, has a negative charge. So all DNA molecules are going to move from the negative electrode towards the positive because opposites attract. But what happens is the DNA doesn't move at the same speed. The smaller pieces of DNA are going to move faster than the larger pieces of DNA. And therefore, when you turn the gel off, you'll be able to see, for example, this band right here of DNA is smaller than this band because it traveled further. So gels are used for many different purposes, and when we get to the biotechnology chapter, we'll talk about those more, but I just wanted you to see the basic idea that they're separating DNA based on its size or its length.